All right, then I'm going to go ahead and all right, so yesterday when we, rather on Monday, excuse me, on Monday on our last class, when we finished lesson 3, we moved straight on into lesson 4 and went ahead and discussed enzymes. So that makes today, actually, we are going to have a very short lesson. We've already discussed much of the material for lesson 4. We talked a little bit about how enzymes work to catalyze the reaction that takes reactants to products, and how in the process of catalysis, the enzyme itself isn't used up. Rather, it just gets, it's available to be used again and again in this cycle. We also talked about the induced fit model, how when a substrate comes to bind to an enzyme, it actually alters slightly the binding pocket on the enzyme where the substrate binds, and it induces a better fit, and therefore creating this enzyme substrate complex. And then that positions all the residues of around the binding pocket on this enzyme to catalyze the reaction that takes the reactant to the product. In this case, the reaction is the hydrolysis of a disaccharide. In this case, it's sucrose. And through hydrolysis, it gets broken down into its product of glucose and fructose. And at that point, the products leave the active site, and the enzyme's active sites are available again for another round of binding to the substrate, catalyzing the reaction. The products leave, and the cycle goes on and on. We had a discussion about how various factors such as temperature and pH affect the rate of enzyme catalyzed reactions. And we discussed that there is always an optimal pH and optimal temperature at which the rate of the catalyzed reaction is at its maximal point. And if you increase the temperature beyond that point, the rate of reaction then rapidly drops. And this is true also for pH. There's an optimal pH where you have maximal rate of reaction, and beyond that pH, either below or above that optimal pH, you have a decrease in the rate of the catalysis for the reaction. And eventually, that is as a result of the enzyme being denatured, because if you increase the temperature or you increase the pH or decrease the pH, what you're doing is you're affecting those intramolecular bonds and affecting the structure, the three-dimensional structure of the protein. And the three-dimensional structure of the protein is very, very closely tied to its function. So if you disrupt that three-dimensional function, you have in fact disrupted three-dimensional structure, you have in fact disrupted its function. And then we discussed the other two factors that affect the rate of the reaction, and that is the concentrations of the enzyme. So if you have more enzyme present, obviously you're going to have be able to catalyze more and more of the reaction, taking the substrate to the product. So you have an increased rate of reaction. However, if you increase the substrate concentration, you do eventually increase to a point of reaching a point of saturation. Increasing the substrate concentration beyond that does not affect the rate of the reaction. And that is because at that point, no matter how much more substrate you add in, the enzyme's active sites are already saturated and bound. And so simply adding more substrate no longer affects the rate of the reaction. We talked a little bit about enzyme inhibitors and activators. Enzyme inhibitors are the type of molecules that can bind to the enzyme and decrease its activity. And conversely, enzyme activators are the ones that bind to the enzyme and actually increase that enzymatic activity. Then we discussed the few different ways that the substances that may bring about inhibition or activation can actually affect the rate of the reaction of the enzymes. And to understand how this inhibition actually works, we have to understand the structure of the enzyme and be aware that oftentimes on the enzymes, there are two different distinct binding sites. One is referred to as the active site, and the other important site on an enzyme would be an allosteric site. The active site is usually a patch or a pocket on the enzyme that is surrounded by the amino acids and side chains that have residues that are responsible to allow the substrate to bind, to recognize the substrate, and to catalyze the actual reaction. The allosteric site, however, is in a location other than this active site, and the binding to this allosteric site can have the function of either activating or inactivating the enzyme, again, through binding and either perhaps blocking the access of this substrate to the active site, or perhaps the binding of the inhibitor or the activator to this allosteric site somehow affects and alters the conformation of the enzyme at the active site to allow better or poorer binding of the substrate, and therefore either increasing or decreasing the rate of catalysis. And so historically, there was the lock and key model proposed as to the model that allowed substrate to bind to the enzyme. And the lock and key model basically assumed that the active site was present and it was providing just the perfect fit for a specific substance, just like you would have a very specific key that is made to fit in perfectly into a lock. But in the induced fit model, the assumption is that the active site is somehow, somewhat of the right conformation to be able to bind the substrate. But in fact, the binding of substrate actually induces an even more perfect fit in this site, in this region where there is the interaction between the substrate and the active site. And as a result of this conformational change that occurs, then the enzyme is better able to bind to its substrate. Now, there's different types of inhibition. There is the type of inhibition that is referred to as a competitive inhibition, and similarly, there's also exists non-competitive inhibition. So what do we mean by competitive inhibition and non-competitive inhibition? In competitive inhibition, in this case, we have an inhibitor that seems to be so similar to the actual substrate. So for example, here, this blob here is supposed to represent the inhibitor. And this blob here represents the substrate. So you can see that this inhibitor is very, very similar to part of this substrate, that it's able then to actually enter the enzyme's active site. So instead of the substrate being able to bind at the active site, this other molecule that is not a substrate can bind in the active site. And by doing so, it can stop and block the access of the substrate to this active site. And so if the substrate is not able to enter the active site, the enzyme is not able to catalyze that reaction. So in effect, this inhibitor, this molecule is acting as an inhibitor and stopping the enzyme from catalyzing its reactions. So in this case, because it is binding to the substrate binding site, it is a competitive inhibitor because it is competing with the substrate for the binding site on the enzyme. And in this case, you can reverse this inhibition simply by increasing the amount of the enzyme's regular substrate. So essentially, if you have more of the substrate molecule, then perhaps it can outcompete the competitive inhibitor and have more chances of being able to bind into this binding site. So now if we try and imagine what's the situation in non-competitive inhibition, in non-competitive inhibition, you have a molecule that doesn't compete for the enzyme's active site where the substrate normally binds. Instead, this other molecule, this type of inhibition occurs at the allosteric site, the site that is somewhere else other than the active site. And the binding of this molecule then to the allosteric site causes a change in the shape and the conformation of the enzyme overall, causing it to alter its active site and stops it from being able to bind to its regular substrate. So now, as you can see, the regular substrate can no longer bind to its active site in this pocket. This pocket has now adopted a different conformation that is not so conducive to the binding of this substrate. So in this case, we say that this is a non-competitive inhibitor because this molecule is not actually competing with the substrate for the same site on the enzyme. It is binding at a different site. So this type of inhibition is non-competitive. And sometimes this binding and enzyme's conformation then is, this binding is so tight that this induces a permanent change in the conformation and in the three-dimensional structure of this enzyme at its binding site. And in this case, then this substance that is bound to the enzyme oftentimes acts as a toxin because it is binding so tightly and it is not coming off. And so it acts as a toxin to the organism. So most toxin molecules are the type of molecules that bind and bind quite tightly and permanently change the substrate binding site on the enzyme. Now, this binding of inhibitors, as we discussed, can sometimes be reversible, so it can come on and off, or it can be irreversible. And in the case of a molecule that is binding irreversibly and binding quite tightly, usually this binding causes a reaction with the enzyme that actually changes it at a chemical level. It actually changes and alters this enzyme in a way that it no longer is able to bind. It's caused the modification of some of the amino acid residues that are important to the enzyme's activity. In contrast, when you have a reversible type of inhibition, this type of binding does not change any of the amino acid residues. It's non-covalent type of bonding, and depending on whether the inhibitors bind the enzymes, then this enzyme substrate can be both reversibly inhibited or irreversibly inhibited. So as we discussed, there's that allosteric site, a site that's different than the actual active site. So this means that that allosteric site can be used as a way of regulating the rate of the enzymatic reactions. And cells control their enzymatic pathways in order to be able to provide a level of coordination within the cell of all the different reactions and pathways and activities that are going on in the cells. And cells achieve this in a couple of different ways. One way is to actually limit the production of a specific enzyme. So if you don't produce as much enzyme, well, not as much of that reaction that is catalyzed by that enzyme can take place. Another way they do this is to inhibit the action of the enzyme that has already been produced. So if there's already enzyme produced, another way of doing it is to inhibit it through the use of these inhibitor molecules. And so molecules that cause an increase and promote the activity of an enzyme and do so by binding to the allosteric site of the enzymes, those are known as activator molecules. And when you have the accumulation of a substance within a cell, then a cell may then specifically activate an enzyme by binding to its allosteric site, and as a result, causing a new sequence of reactions for that activated substance to the initial substrate. And in this type of regulation, this is called precursor activation. Other molecules can act as inhibitors of the enzymes. So sometimes you have drug molecules. Many drug molecules, for example, are enzyme inhibitors. And actually, research and development in this area is quite active. And the utility of an enzyme inhibitor is often judged by how specifically it is able to bind to its target location. So its specificity, or sometimes the lack of its specificity of being able to bind to other proteins, and also its potency. How potent is it? How strongly does it bind? And also, when you have a drug molecule with high specificity and potency, that is a way of ensuring that that drug will have very few side effects because it targets very specifically. And so therefore it will have low toxicity. It doesn't affect everything in the cell. It is quite specific to a certain enzyme that is catalyzing a certain point in a metabolic pathway. So that is in the case of when you have a drug molecule, but also naturally, enzyme inhibition is a way for the regulation of cell metabolism. For example, you have enzymes in a metabolic pathway that can be inhibited by the product that is produced further along the pathway, further downstream of the pathway. And when you have this type of pathway, this type of regulation, this is called feedback inhibition, because you have the product that is coming downstream later on in the pathway, feeding back upstream and inhibiting that enzyme from producing more of it. So that this is called a feedback inhibition. It slows down the pathway so that a specific product doesn't build up in the system. And feedback inhibition is a type of non-competitive inhibition in which the end product of that series of enzymatic reactions binds to the allosteric site on the first enzyme that was part of that pathway. And this is one of the ways that the cell is able to maintain homeostasis, which is a way for the cell to balance its internal processes. And we will be talking more about homeostasis later on in our Unit 4. And also just to note, there are other natural enzyme inhibitors that can act as poisons, and these are often used as defense mechanism, as a way for an organism to defend itself against predators, or as a way for an organism to kill its prey. So a lot of these inhibitors and a lot of these poisons are also enzyme inhibitors. So here's a diagram that demonstrates that concept of feedback inhibition that we talked about. So let's take a look at exactly what's happening over here. So let's see that here we have a series of steps. So this is a pathway in which a series of reactions occur. And it starts out initially with an initial substrate. So this is the first reaction that takes this initial substrate. In this case, it's a molecule of aspartate. And through this reaction 1, it goes to produce a certain intermediate A, which then is catalyzed through the action of this reaction 1 is catalyzed by this enzyme 1, and produces this intermediate A. And then intermediate A is catalyzed by enzyme 2 to produce this intermediate B, which is then catalyzed by enzyme 3 to give this intermediate C, and so on and so forth. So as you can see, there's different enzymes, 5 different enzymes along this pathway that catalyze all the different reactions that take place. So, and finally, the end product of this, this pathway is to produce a molecule of threonine. That is the end product. So initial substrate was aspartate. It went through a series of reactions and intermediates to produce finally the end product threonine. Now, the end product threonine has the ability to bind at an allosteric site on enzyme 1. Enzyme 1 being that enzyme that catalyzed the first reaction of taking that aspartate. So if you can see this active site here, where that's rectangular, let's say, obviously in life, this is not rectangular. We're just looking at this as we're just using some geometric shapes to represent the different intermediates along the pathway. So let's say this molecule that here we've represented as a rectangle can fit nicely into this active site here. And so when it binds, it then gets catalyzed and produces this intermediate A. But this threonine here is able to bind at an allosteric site. And through the binding of this molecule at the allosteric site, it can distort this active site such that it can no longer bind this substrate. And so as a result, the binding of this end product of the pathway to this upstream enzyme shuts down the pathway. It doesn't allow this pathway to go on anymore. So in the case of this feedback inhibition, this causes the shutting down of the pathway. So it's important to note that in this case, the activity of the enzyme can be regulated not just by its binding of its substrate, but also by a molecule that is produced later on and downstream of the pathway that is not its substrate. And in both of these cases, you have this regulator molecule that is binding to the enzyme at a different location than the active site. So here again, there are some examples of allosteric regulation. So in this case, you have the enzyme that has an active site. And for example, this enzyme might exist as as 3 subunits that come together as 3 units of this enzyme. And when they come together, you have this allosteric site here. And this is the active site. And when you have a binding to the allosteric site, it maybe changes the conformation in a way such that you have an inactive form now, and it can no longer have the proper shape at the active site and does not allow the binding of the substrate. But then sometimes you might have a molecule that would act as an activator, and the activator then binds to the allosteric site and stabilizes this active form such that it is able to bind to its substrate molecule, and it doesn't seem to adopt this inactive form. So in this case, in the presence of the activator, the active form is stabilized by this allosteric activator, and the inactive form would be stabilized by a molecule of an inhibitor molecule. So if you can imagine this enzyme that can adopt both of these conformations, an active conformation and an inactive conformation, in the presence of an activator, it is the active form that is stabilized. But in the presence of an inhibitor, a molecule that acts as an inhibitor, what that molecule is doing is stabilizing a form that cannot carry out the reaction. So I have actually it occurred to me that really sometimes the best way to look at proteins and enzymes and to get an appreciation for what we mean by 3-dimensional structure, what do we mean by this pocket that is lined with amino acids with the right residues and the right functional groups in place. What do we mean by that? Sometimes it's a good idea for you guys to be able to see that better. Over here, we're kind of representing these very abstractly and kind of as a flat image. I have some videos lined up that maybe might be useful to you. And since we have time today might be a good time for me to share some of those videos with you, just so that you could have an appreciation of how these how enzymes and substrates bind. So let me just pull that up for you. So first of all, I want to maybe show you the video that discusses what is a protein. That might show you a better picture of what is a protein. So let's have a look at that.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom.
So this is something that we discussed in our previous lesson. We talked about the different amino acids and that they all have this central carbon atom, and bound to it is a carboxyl group, an amino group, a hydrogen, and then there is this side chain.
The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid.
So here, if you look at the different amino acids, they've highlighted how each one of them has a side chain that is different. So an alanine will have this type of side chain, a leucine will have this type of side chain, phenylalanine will have this type of side chain, so on and so forth.
Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic or polar amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules.
So this is another concept we discussed. We discussed that there are amino acids that are hydrophobic, and that's because, as you can see, their side chain is made up of mostly of hydrocarbons, and so it doesn't like to interact with water very much. Then there is a group of amino acids where the side chains are quite polar and hydrophilic. They like to interact with water. Then you have amino acids whose side chains are charged, have got a charge on it. And so therefore, they are quite happy to interact with molecules that present the opposite charge to what they have. And this dictates where they occur in the overall three-dimensional structure of a protein and what kind of a reaction they might help to catalyze.
Primary structure.
So we discussed also that their protein structure has got these 4 levels, this hierarchy. You have the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. And here in this video, you can see the animated view that I am not able to show when I'm just talking about these concepts with you in static pictures. So I thought it might be helpful for you to see this also.
Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into 2 types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize 2 or more adjacent strands of amino acids. The tertiary structure of a protein is the 3-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain.
Right, so you have this long chain of all the different amino acids as determined by the primary sequence. And the primary structure, the primary sequence, it's just the order in which what amino acid comes after what. And then those amino acids might get folded into those secondary type of structures. There might be some sheets that are stabilized across due to hydrogen bonding. You might have a piece of a the polypeptide that might form this coil called this alpha helical coil over here. So, and then overall, they all come together and are stabilized. And obviously, proteins are in an aqueous solution. So you would have the hydrophilic residues pointing outward, and the more hydrophobic residues and side chains buried into the center of the protein. So here you can see the hydrophobic ones are the ones that are pink, and you can see there's more of them in the center of the protein. Yeah, there's some on the surface here too. And there are some charged amino acid groups, those yellow ones, and then there's the hydrophilic, the blue ones that are facing outside and are happy to interact with water.
Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors.
Right, so this is that lipid bilayer that we discussed with the polar phosphate head facing the outside of the two outer sides of the membrane. And then the inner part of the membrane is where those lipid chains that are quite hydrophobic like to hide away from water. And then sometimes you have these membrane proteins. They might be channels or transporters or pumps, and they are embedded in the lipid bilayer. And parts of it might be actually crossing and traversing the lipid bilayer. So there might be part of it that's facing the inside of the cell or outside of the cell. And in that case, you see that in the lipid bilayer, where the amino acids are protected from interacting with water, is exactly where those hydrophobic amino acids like to concentrate. Oftentimes, the part that's actually passing through the membrane is lined with hydrophobic amino acids.
So that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds.
So did you see how that pocket allows the binding of its substrate? So that's what we mean about how the three-dimensional structure has got a lot to do with the function of the protein. Disrupt this three-dimensional structure and you're disrupting the function of the protein, because then that protein will no longer have the proper shape.
Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas on the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Data Bank.
All right, so I just wanted to show that to you. As a way of just enriching your learning and opening your eyes to proteins are so much more, and you can see the huge variety of proteins. And again, just to highlight how important the three-dimensional structure of a protein is to its function, whether it be enzymatic, whether it be structural. It's very closely tied together. And then when you have time and if you're interested, this video that I showed you is from the Protein Data Bank. They have put together these videos that are so instructional. And if you're just wondering what is the Protein Data Bank, I can also show you that. So if you want to see. All right, so this one, I'm just going to play a little bit of it just so that you can have an idea about the Protein Data Bank. And you can, if you like, you can go to their homepage and explore and play around a little bit with the resources there. It's fascinating.
The Protein Data Bank Archive is an online repository of 3D structures of proteins and nucleic acids. All structures in the PDB were experimentally determined, and for every structure, the PDB contains the X, Y, and Z coordinates of each atom. RCSB PDB is one of the organizations that manages the PDB archive. It makes the data publicly available on rcsb.org. where it also provides tools for query, analysis, and visualization.
So you can go to rcsb.org and look up various proteins that you might be curious about, and you'll see the wealth of information that is available at this database site, where you can find out so much about these proteins whose structures have been determined.
RCSB PDB also maintains an educational portal, PDB101, accessible to everyone free of charge at pdb101.rcsb.org. PDB101 offers a diverse array of teaching resources highlighting the fundamental connection between the 3D structure of a protein and its biological function.
Again, that is key. The three-dimensional structure of a protein is fundamental to its function.
The materials are organized by type, accessible through the top menu, and by topic, accessible through the browser. One of the key resources on PDB101 is Molecule of the Month. Each column focuses on one structure from the PDB archive and includes an introduction to the structure and function of the molecule, a discussion of the molecule's relevance to human health and welfare, and an interactive viewer enabling exploration of relevant molecular features in 3D. Under the Learn tab, PDB101 features various resources that can supplement any curriculum with 3D structure-driven activities for all types of learners. Looking for hands-on learning resources? Bring PDB101's hands-on paper models into the classroom. Building these models will help students to become familiar with key aspects of protein structure. Additionally, PDB101 provides 3D printable models that highlight interactions between a protein and its ligands. PDB101 also features molecular animations explaining general topics such as what proteins are and how enzymes function, as well as more focused topics like pain signaling and ion pumps. PDB101 also contains interactive animations that enable students to digitally explore the world of proteins. For example, the interactive molecular machinery poster provides a proteomic tour from the cell membrane to the nucleus, offering 3D views for each of the proteins featured. Looking for a class project where students can combine research and science communication? The Video Challenge for high school students is an annual event where students research a medically relevant science topic and create a 2-minute video combining the topic's molecular and public health aspects. PDB101 also offers a variety of comprehensive curricular modules under the Teach tab, covering topics ranging from biomolecular structures and models to molecular immunology. These modules are fully outfitted with learning objectives, hands-on activities, and before and after tests that enable teachers to assess student understanding. Finally, if you would like to bring advanced 3D protein structure visualization into the classroom, the interactive MolStar viewer is available on rcsb.org. The MolStar viewer is accessible from the structure summary page of a given protein entry in the PDB archive. A user guide and video tutorial are also available for the MolStar viewer. Please visit rcsb.org and pdb101.rcsb.org today to begin your journey.
So that I just wanted to share that with you so that you understand that there are some of these resources that are available to you if you want to go ahead and satisfy your curiosity and find out more about these various proteins. So finally, I want to share one more video with you since we have time, and that is again from RCS PDB 101, and this one has to do with how enzymes work.
Into the world of proteins.
And I'm just going to bring that over. And this should help you visualize how enzymes work, which is what we've been talking about.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy, carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize a transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction, citric acid cycle, that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitase. It then adds the hydrogen and the hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl and substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect.
Yeah, so see how there's quite a lot of flexibility in proteins. They're not rigid. They're quite dynamic. There's motion and movement in it. And you can study. There's protein dynamics is an area that is very much studied by scientists to better understand how these enzymes and proteins function. As you can see, this part of this protein, this pocket here, has got a little bit of a wiggle room. It opens and closes to allow entry of substrate in there. And this is where that induced fit comes in. So let's have a look at that again.
Environment for a reaction.
Just gonna
Around their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell, in every living creature on the planet.
Great. So I just really wanted to share those videos with you because I hope you guys also found it useful to see that view of enzyme substrate binding and that view of proteins and view of the residues that are present in the active site of these enzymes. And from that, you can imagine how binding of a molecule to the allosteric site may ultimately affect the overall shape of an enzyme in a way to alter the conformation of this active site and perhaps not allow the substrate to bind, or to hold this protein in a conformation that very much allows this substrate to bind and maintains that active conformation. Now let's take a look at what are some of the uses of enzymes. So enzymes can have, other than, of course, within organisms, as you could see, they catalyze thousands of reactions in metabolic pathways, and that's certainly one role that they have, but they also can have many applications in commerce and in industry. So for example, you can have enzymes that are proteases, and they are very useful in the dairy industry in the production of cheese. And these proteases, also together with enzymes called amylases, are oftentimes added to detergents, and that will help the detergents be able to remove stains that are present from proteins and carbohydrates, and to help to break down those stains that are have that kind of carbohydrate or protein nature. These proteases and amylases are then able to break those molecules down and help to remove the stains better. So there's tons of industrial uses for enzymes. For example, enzymes that have the ability to convert starch into sugars, and then when the sugars are then converted to ethanol. So that's enzymes that are involved in fermentation. So in the production of alcoholic beverages and wine, these enzymes are carrying out that reaction. There are, in baking, for example, you can have flour that is modified with enzymes to make it have improved baking properties, might be lighter, might be fluffier. Again, in dairy, as we mentioned, in cheese making. And we talked about in detergents. So there's excuse me, there's many uses for enzymes in commercial applications and in industries. Finally, we have to wrap up our discussion with a discussion of coenzymes. So sometimes enzymes need some non-protein cofactor to help them carry out their function. And some of these cofactors are ions, for example, like magnesium, like calcium. Others are called coenzymes. And these coenzymes are inorganic molecules that can bind to enzymes and help as carriers of chemical groups or provide electrons, for example. And we'll be taking a look at these important coenzymes of NAD+ and FAD, for example. These are very important coenzymes when we take a closer look at cellular respiration. Many coenzymes depend on our intake of vitamins. For example, NAD+ is dependent on the vitamin niacin, and FAD is dependent on the vitamin riboflavin. And if you have a deficiency in one of these vitamins, it may result in a lack of a coenzyme, and so then you will have a decrease in a certain enzymatic reaction. So typically, if you have a very varied diet, you have the ability to obtain all your various vitamins, which will provide you with all the coenzymes and cofactors that are necessary for enzymatic reactions. However, sometimes if people have a very limited diet, or if their diet is deficient in certain nutrients and macromolecules, then they might develop a deficiency that may require them to take vitamins in order to supplement the presence of those coenzymes through taking vitamins. All right, so any questions? I'm wondering, has anybody had has had a chance to look at any of these homework questions? And if there's homework questions that you would like help with, we have some time right now. I'm available. We can go through the homework questions if anybody has any questions, if anybody's attempted those. Any questions? We can use the remaining hour if you guys like. Since we finished our unit, we can use this remaining hour as either a review, or you guys can independently work on your homework questions. And if you have any problems, then we can discuss it as a class. So would you guys like to do that? Would you guys like to try some of the homework questions, and then we can perhaps take it up as a class? All right, well, if anybody, hang on, let me see, there's a message from somebody. What would you like? Okay, so yeah, anybody who wants to can stay and work on homework. And if you have questions, I can help you. Anybody who feels like they have to, they would like to leave the lesson, you can leave now, and we can meet again next Monday. Okay, so I'm going to be here. I'm just going to mute myself, and I'm going to stop recording.